Okay, we're going to take a look at sample variability again and sample size because it's a very, very important concept around understanding what you're going to be talking about and the conclusion that you make and so on. So what we've got a population. I think it's 400 or 500 people here that have been measured and their heights have been taken. And so we can see a dot plot and a box plot for the population. And what we're going to do is we're going to separate this population into gender, male and female, and look at their height. So we want to do a comparison. Now, in the real world, we don't know what the population is. We don't look at the population because we don't have the entire group of individuals to look at. But if we did, we would see that from our population, software can then find the medians of the males and find the medians of the females. And although we don't see what actually these medians are, we can see that the difference between the males and females' average height is small. There's not much difference between them at all. It's about 2 centimeters. But the fact of the matter is, under normal circumstances, we don't know what's going on in the population. So what we do is we randomly pull out a sample from the population. We create a sampling method so that we can randomly select some individuals and we pull them out of the population and we look at that and we draw a conclusion based on our sample about what's going on in the population. So let's say we pull out 60 individuals at random from the population and we create a dot in a box plot and we have a look at what's going on in the sample. Now, if you remember, we had in our population a difference of 2 centimeters, and that was the position of the median and so we took out a sample. It looks like that's pretty close to what's going on in the population. So that's actually quite a good one. Let's take a look at another sample. So we'll pull out another 60 people at random out of the population, and we'll see what we get for that one. A bit of a difference. Position of the medians is a little bit different, but it's fairly similar. Let's take another one. Remember, the sample has to be taken randomly so that every individual has an equal chance of being chosen from the population, and then we drop it down. Now, you see this sample? This could be Sally's sample. Now, look at Sally's sample. Sally's sample looks quite different from the other 2 samples, and it certainly looks quite different from the population. So if we don't know what's going on in the population and we rely on Sally's sample to tell us what's going on back in the population, we could be incorrect. We could be wrong. There's a really strong case here for the fact that Sally's sample doesn't tell us accurately what's going on back in the population. The first 2 didn't seem too bad. The third one, not so great. Let's take another one and see if Mary's sample that she's taken out of the population is better. Well, suddenly we see that when we go back into the population, we saw that the females tended to be a little bit taller, not by much, but by a little bit. Here, the males are taller. Completely different story. Now, there's not much difference. So that's why we do this analysis around how much overlap there is between the samples and the box plots and where the position of the medians in. And then we look at overall visible spread and so on so that we can kind of start to make some conclusions about the 2 samples. And so we can kind of make some inferences and conclusion about what's going on back in the population because we won't see what's going on in the population. We have to base our analysis around a single sample. And that's a problem because sample variability tells us if I do another sample, it's going to be quite different from my original sample or another sample or Mary's sample or Tom's sample and so on. So every time we do a sample, we get sort of an idea what's going on back in the population, but it's not a complete picture. So there's a lot of variability. The other thing that goes on with variability is that the bigger the sample, the less variability you will get between groups and between samples. So, for example, if I took a sample of 10, which is a really small sample, it means that there's a lot of variability. These blue lines that you see are medians for there's like thousands of samples being taken here by software. And the blue lines are the medians. So the medians are shifting, shifting, shifting. And you can see that the box, its position and its length and so on are shifting, shifting, shifting. And it's a big shift. It's a lot of variability here. If I take a sample size of 100, all that shift becomes a little bit less. You know, the boxes move less. So these red lines are a little bit narrower. The median, the blue, is less variability in that, and it doesn't move as much as it does in the 10. And then if I was to do a sample size of 1,000, you can see that it starts to mirror what's going on in the population. So this is a better idea. We get less variability because the more individuals we have, the more it's going to mirror what's going on in the population. So sample size is important as well. So you want to take a good, robust sample. If you take a sample of 10, that's going to be very difficult to draw some conclusions about because every time you do a sample, they're a lot different from each other. If you take a sample size of 100, then the sample that you have against somebody else's sample, there'll be less variation between those samples. And if you take a sample size of 1,000, if Mary takes a sample and I take a sample and Tom takes a sample, there's going to be less variation between those samples. So we can kind of feel better about what we're saying about our sample back in the population because it's sort of mirroring what's going on. So those concepts are quite important for your understanding.